I wanted to thank everyone for coming, and I wanted to really thank the organizers for this really fantastic meeting. This is my third opportunity to present at HFSP meeting, and this has rapidly become one of my absolute favorite conferences. Really enthusiastic about the science, and it's a really great opportunity to present some of the work. Secondly, a confession. So I'm an alumnus from HFSP, but the work I'm presenting is actually not the work that was funded by the HFSP, but the organizers kindly let me present this work, and I hope you enjoy it. And then the third part is I'm going to give a 15, 20-minute talk, which is presenting the work of my postdoc, Balu Balan, which he started his PhD, and it's sort of 6 years of his life that you're going to get in 15 minutes, but he's done a tremendous amount of work, and we're getting quite close now to a point where I think we're going to be ready to publish it. Okay, so we've heard a lot at this meeting already about the role of RNA binding proteins. This is an area that we're particularly interested in. We're interested in how these systems regulate the biology of eukaryotes. And I'm not going to go into detail on all of these different ways in which this happens, but one of the things we can take away from the radiation of eukaryotic organisms is that this task of separating a nucleus from a cytoplasm and compartmentalizing transcription from translation created a significant regulatory challenge, which is this massive layer of regulation. You need to be able to get RNA out of the nucleus, into the cytoplasm, these things. But it also created a tremendous evolutionary opportunity, which is that you added a fantastic distance between transcription and translation in which many different layers of regulation could evolve. And this became hugely important in a lot of the complexity that we see in eukaryotic cells. In addition to these sort of functions that you may be familiar with, transcription, translation, splicing, RNA decay, etc., as we've heard a little bit at the meeting as well, RNA-binding proteins have this fantastic capability of forming these complex RNA protein meshes that are referred to in many different terms: dense granules, membranous organelles, liquid-liquid phase separation, condensates, whatever. Little gel balls that are RNA and protein and probably some other things that are distinct within the cytoplasm that allow for subcellular regulation and localization. And they allow the cell to do a lot of different things. They can repress transcripts and hold them for later. These are really important in programming cell fate. You name the major developmental phenotype, and I'll be willing to bet that there's a lot of RNA-binding proteins involved. And they can orchestrate complex RNA regulons, where you have RNA-binding proteins binding many hundreds of RNA molecules, many of those RNA molecules themselves being RNA-binding proteins that, when released and translated, have further regulatory networks that they control. These are known to play fantastic and important roles in a wide variety of aspects of higher eukaryotic cellular life, including in patterning embryogenesis, cell development, development of immune systems, so on. Much of this will be familiar with many of you already. But it also raises a lot of interesting questions, and not surprisingly, I have a whole lot of them. I'm not going to read them all, but you can guess the basic gist of this. We want to understand where these systems came from. RNA-binding proteins exist across all cellular life. RNA-protein interactions are predicted to be maybe one of the things that exist prior to cellular life, and arguably one of the most important macromolecular interactions that lead to life. But the systems that prokaryotes have and the systems that eukaryotes have a lot of distinctions. And there's a huge amount of RNA biological control that evolves in eukaryotes that do not have an analog that we can identify in prokaryotic life. So that begs the question, well, where did they come from? And then we can ask many subsequent questions. If you imagine any question you want to think about in terms of how these systems evolved, we can ask these types of questions. Where did they radiate? What did the proteins look like in their simpler version? How did they gain greater complexity, and so on? You can also ask interesting questions around the differences in the relative importance of different layers of epigenetic control. RNA-binding proteins versus chromatin control versus transcription factors. Am I interested in RNA-binding proteins because they're important, or RNA-binding proteins important because I'm interested in? Well, probably a bit of both. But so we thought we might try and contribute a little bit to this conversation. And when people had looked at this, because others have considered this, and they looked at different layers of regulatory control that evolved in eukaryotes, one of the things that stands out about RNA-binding proteins compared to, for example, transcription factors and chromatin condensation is that though those latter two layers of regulation are also ancient within eukaryotes, when you look from yeast through to humans, you see that it's really only fairly high up, or plants, really high up in the canopy of the eukaryotic tree, relatively speaking, that you start to see this radiation of complexity. At the basal parts of the eukaryotic tree, those systems exist, but they're fairly simple. So you can see where this complexity arose. But with RNA-binding proteins, whether you look from yeast all the way through to humans across 1 billion years of evolution, pretty much all the systems are already there. They get a little more complex in humans, but they're there. So that begged the question, well, okay, if they're not in prokaryotes and they're in yeast all the way through humans, where might else they be? Well, protists, right? That seems a place to look. So as I said, we thought we might contribute to this conversation. My lab, among many things, works on a parasite called Giardia. which I was pleased. Yesterday I saw it show up in someone else's talk, which is nice. And we're interested in this as an infectious agent of disease, but we're also interested in this because it sits within a really deep branching lineage. I'm not going to try and point. You're just going to have to take my word for it. In the metamonads, they're right down at the base of the eukaryotic tree of life. And so we thought we might be interested in looking at these. And this isn't the first time. So Giardia has some interesting attributes. It's been studied maybe 20 years ago because it doesn't really have a mitochondria. It has this kind of non-canonical mitosome, which probably doesn't actually have an origin from a mitochondria. And so this got people interested in the idea of eukaryotes that predated the evolution of mitochondria. Doesn't have a Golgi body. It has some interesting attributes to it. And that's not just Giardia, that's the metamonads that it fits within. Giardia is a parasite, but other non-parasitic versions within that lineage, similar type of thing. So we've asked some questions around this, and we thought, well, if we wanted to look at some of these questions around the regulatory control of an organism and look at a basal layer, maybe this is a nice system we can work on. We can grow it in the lab as much as we want. You can get it in jars this big if you'd like to, in cell-free media. We can manipulate it. CRISPR is doable. It's not trivial because it's a tetraploid, but it's doable. And it's also interesting in the context of thinking about the importance of RNA-binding proteins and their regulatory systems because Giardia is a bit weird. The metamonads are a bit weird among eukaryotes. They have histones. They do epigenetic modification of those histones, but they have one of the loosest condensomes of a known eukaryote. They don't really use chromatin condensation all that much. They also have transcription factors, but they don't have a really complex repertoire of transcription factors. And it seems like they transcribe most of their genes most of the time. And rather, they seem to regulate their expression of proteins through post-transcriptional regulation. The classic or most well-known example is Giardia coats itself with a protein called a variant surface protein, because it's on the surface and it's variant. We're really imaginative in Giardia field. It encodes about 300 of these proteins in its genome. It transcribes all of them. This was a paper that was published in PNAS about 15 years ago. It transcribes all of them, but it only expresses one at any one time on the trophozoite surface. And it does this through RNA silencing, which is the most inefficient way I can imagine to do this process. It makes zero sense at all. And the only thing that I can think of is, well, this is what it has available to it. So this is what it does. Okay, so it doesn't really have chromatin control so much. It doesn't really have transcriptional control so much. Well, what about its RNA-binding protein systems? So we started to look at that, and this is what Baloo focused on his time. So to do that, we kind of got really excited about RNA-binding proteins, and so Baloo went about mapping, and as far as I know, this is the most comprehensive map for RNA-binding proteins that have been assembled. This is condensed down into major orders, but this represents about 16,000 species across the tree of life that we've done in silico-based annotation of RNA-binding proteins and domains. You create hidden Markov models from known RNA-binding domains. You use these to scan the genomes of these organisms. You do a series of curatorial steps, and you classify them into groups. And I'll take a moment to make some observations about this. And I'm sorry the print is maybe a bit small, but closest to me, we have domains that are across all cellular life. These are lots of ribosomal proteins, RNA polymerases, things like this. Out of the 75 genes that are known to be essential for what they refer to as the minimum synthetic cell, I think 60 of these are RNA-binding proteins, and they sort of fit in this category. So lots of things that are there all the time. Then we've got a series of RNA-binding domains that seem to be bacterial origin. And these would not be surprised. They show up in eukaryotes, in our organellar systems, in mitochondria, chloroplasts, things like this, endosymbiotic theory kind of idea. Then we have a series of domains that were inherited by archaea, and likewise with the idea of the origin of eukaryotes, these tend to be associated with the nucleus. That makes sense. Some of the early splicing proteins, archaeal proteins, things like this. And then we have all of these eukaryotic emergent domains, and these are the things that you'd associate with eukaryotes: mRNA processing, splicing systems, capping systems, transport, RNA decay systems, RNA interference, and so on, translational repression systems, and all the way through. And what we see in this is right down where the metamonad is, where my little happy Giardia trophozoite sits, we see that it has all these complex ones, and it has a lot of, but a reduced number of, the higher eukaryotic function. So it does seem to fit within that kind of transition point idea. It's like higher eukaryotes, it's got a little bit of bacterial stuff too, but it's simpler. Am I halfway yet? Yep, okay, cool. Good. Okay, so we can break down those domains into different groups. We can see the sorts of things that emerge. We see that Giardia starts to gain a lot of the, so RRM motifs. These are splicing proteins, RNA recognition motifs. These arise in eukaryotes. We see them showing up in the first time in Giardia. We see puff proteins. These are really important in cell fading in many higher eukaryotes. Again, these are unique to eukaryotes. We see them showing up for the first time in Giardia. Giardia was one of the first organisms in which dicer protein was described and crystallized with a science paper around 2001. And lots of these other systems appear present. And when you start to then map these across these different functional units, you see that Giardia has all the stuff that it needs to get its mRNAs out of a cell, to transfer. It doesn't do a lot of splicing, but it does have a couple introns, and to do the sort of translational repression systems and these things. But this is all in silico based. So then we started to try and see, well, could we prove it? So we did an experiment, Baloo did an experiment called PowerClip, where you cross-link the protein and RNA within a live cell. You then lyse the cell, use an oligo-DTB to pull out any so we're focused on mRNAs in this example, to pull out any polyadenylated mRNAs. You digest the mRNA itself. You take the proteins that are left with appropriate controls to make sure you wash them correctly. You put that on a mass spec, and you see, compared to those controls, what am I enriching in my proteins? What's bound to my mRNAs? And then you compare that to our in silico predictions, and okay, how many of the RNA-binding proteins we said are RNA-binding proteins are actually binding RNAs? And so we see good agreement with the canonical RNA-binding proteins, the ones that have predictable RNA-binding protein domains. The other thing that we see in our data is a lot of non-canonical RNA-binding proteins, and these are known, and a lot of the ones we find are ones that have been previously described, and these exist also in bacterial systems. One of the really interesting examples is in glycolysis. There are about a dozen of the genes of the enzymes in glycolysis have a moonlighting RNA-binding protein function, where they bind the mRNA either upstream or downstream of them within the metabolic pathway, and they provide a metabolic flux capacity where they can suppress their own transcription or activate their own translation or activity as an enzyme based on binding and interacting. But this happens through these intrinsically disordered regions that are the sticky amino acids rather than a canonical domain. So you see these in Giardia, and you can start to see these expand, and these can become quite interesting. We can also ask questions about how the proteins evolve, not just in terms of where they're present, but then what happens if we look at Giardia compared to higher eukaryotes, and what does this tell us? So we see lots of different things happening with our RNA-binding protein systems. We have simple versions like PUF on the far left. I don't know my left from my right, but that's the one. And the Giardia PUF shown here, and sorry if it's difficult to see the blue against the gray, but PUF proteins from apicomplexans all the way up to humans have these stacked arrays of these PUF domain, and they usually have them in a crescent shape of 8 motifs. And this allows them to bind in such a predictable way that based on the PUF sequence, you can predict the motif that it interacts with. But Giardia's PUF is just one of these motifs. It shouldn't function, but it does, right? When we've proven that, because we can pull down PUF and we can see that it's binding the mRNAs. So we see it in its simplest form. The other thing that higher eukaryotic, compared to us, this is yeast in humans as well, is that they start to expand their intrinsically disordered regions. And these in RNA-binding proteins have a lot of functions. One of them is to allow them to increase the capacity of mRNAs that can carry in these RNA regulons in these sort of dense granules. It allows them to have greater saturation. It also gives them a lot more specificity. These get epigenetically controlled through post-translational modifications, and this allows some fine-tuning. And in Giardia, we don't see these. Or we see them, but we see them in a greatly reduced form. We also see addition of domains. Dead box helicases start out with a dead box helicase domain, but as they evolve, they specialize through adding additional domains that are not RNA-binding proteins. We see radiation, RMs, for example, radiate through family expansion. So we see all this across these groups. Touched on this, we see radiation. Not only do we see that while Giardia has these RNA-binding domains that higher eukaryotes have, as you move into up the crown of the eukaryotic tree of life, you see that across the board, you see radiations and expansions, gene duplication events. There's a lot greater complexity as we go up to yeast in humans. So proving this idea that Giardia kind of sits within this simpler nexus point that we can study. And then we can start pulling out individual proteins, and we can start studying them in greater detail. We can ask questions. We've focused on 4 RNA-binding proteins. One is DDX3, which is an important RNA-binding protein regulating, for example, stress responses. Another one is PUF, which is important in cell fading. We look at phosphoglycerate kinase, which is one of the moonlighting glycolytic enzymes. And then we look at E1F4 alpha, which is involved in ribosomal function. We can express those recombinantly. We can develop HA-tagged overexpressed versions in Giardia. We can cross-link them. We can do the reverse process. So we can now pull down the PUF. And we're not the only people that do this, but we're doing it in Giardia. This is not a new technique. We can pull PUF down, and we can do the opposite of that PowerClip experiment. We can then digest the protein, and we can put the mRNA on an RNA sequencing machine, and we can say, okay, what is PUF interacting with specifically? And when we do that, one of the things we see is that, yes, they are, even at this early stage, even in the PUF example with this tiny little domain, they're interacting with a large network of mRNAs. When we compare those mRNAs to orthologs and higher eukaryotes that are known to be regulated by the same protein, we see strong overlap. So even at the very base of the eukaryotic tree of life, these complex regulon networks are already complex. They gain complexity further still, but they're there. I'm running close to time. I'm nearly there. We can also knock down, we can't knock out these RNA-binding proteins, probably because they're just too essential. But we have CRISPR interference that works well in Giardia, and we can knock them down, and we can see what happens. And what we see when we knock these proteins down, well, they translate the protein, the mRNAs that they're binding with, and it's the same network. So that network process is happening. One of the other key questions is, can RNA-binding proteins, even at the base of the eukaryotic tree of life, engage in phase separation? And this has kind of been spoiled now because some colleagues in Sweden have shown that bacteria actually do this a little bit. But nevertheless, let's look at it. And we can also ask questions as we start to define these granules that form in these condensates, is what the minimal composition of these look like in eukaryotes. These are made up not just of one RNA-binding protein, but often a collection of dozens of RNA-binding proteins working together to form these condensates. And this gives a lot of plasticity and programmability into the different types of granules that are forming. And we can show that in Giardia. So what we've done with this, we've taken Giardia, these proteins, we expressed them recombinantly. We worked with our protein production facility at WEHI. Producing recombinant RNA-binding proteins is fiendishly difficult, especially in high purity, but we've developed a method that allows us to produce these at about 90% purity reliably, and are quite excited about that. And this allows us to do some really cool experiments. So you can make the proteins phase separate, you can alter the pH of the salt, and then you can add RNA to the system, and you can see if they form condensates in vitro. And we show that again, has been done by other groups around the world in humans. We've shown it works in Giardia, it does the same thing. So it phase separates. Even with very few intrinsically disordered regions, it still does it. And we're in the process of then demonstrating this happens in vivo. So you can grow the trophozoites, you can label the proteins with a fluorescent marker, green fluorescent protein or cherry, and then you can expose them to, for example, the classic is arsenicide derivatives that cause them to form stress granules. So we can see whether they do that, and then we can see what happens next. We can see how they transcribed across the life cycle, and we can see that at key checkpoints in the developmental cycle of Giardia, they have these major transition points, so it seems to behave like they do in higher eukaryotes. Okay, so what have I shown, and then I'll wrap. I'll finish, I won't actually wrap. So we've shown that both in silico and through in vitro studies that Giardia has, and right at the base of the eukaryotic tree, you have complex RNA biology and RNA-binding proteins, like what we see in higher eukaryotes, but in a simpler form. I think we demonstrate that Giardia is, maybe not the only, but is a suitable model to understand a simplified version of eukaryotic RNA-binding protein regulation as compared to yeast and other eukaryotes above it. And we can follow that through that tree of life and see that complexity is a continuum. When we look at Giardia compared to, say, for example, AP complexins, like for the parasite that causes malaria, we see an increased complexity in malaria in terms of its RNA-binding protein systems as well, across all those different metrics that we look at. We show that nevertheless, despite this simplicity and reduced form, it's engaged in complex RNA-binding protein regulation. It can form membranous organelles, we think. At least we can show it can phase separate. We're doing that last bit now. And these seem to play really important developmental switching. So it seems to function, just like it does in higher eukaryotes, but in a reduced state. And we can start to answer some of these questions a little bit, or contribute to them. So we do have this kind of transition point. We can start to ask questions about how RNA-binding proteins evolved, and we can see that there's evidence of a simpler state. We can also ask questions about the importance of these different regulators relative to the complexity of biology. And I'll conclude there. This is an enormous collaboration. I really want to highlight Balu because this, as I said, is about 6 years of his life. I'd like to thank all of you for your attention. And again, thank you to the organizers for both a fantastic meeting and for the privilege to get to present to you today. Thank you.